All right, I'll go ahead and jump in and introduce our next speaker. Dr. Diana Lin is a senior environmental scientist in the Clean Water Group at San Francisco Estuary Institute. She studies microplastics and other contaminants of emerging concern and legacy contaminants in the San Francisco Bay surface water, sediment, fish, effluents, and stormwater. She and her wonderful team at SFEI just completed the first regional, comprehensive regional survey of microplastics in the San Francisco Bay. And she's going to talk about that and some of the monitoring methods development findings that they had. Take it away, Diana.
Great. Can you see my screen?
Yes.
Thanks. Thank you so much, Shelley. So I will be talking about the field sampling methods and lessons learned from our San Francisco Bay microplastic study. And as Shelley said, this was a huge team effort to conduct the study, so I wanted to thank my colleagues as well as Ezra Miller, who is attending as an attendee, who helped me put together this presentation. So we completed our study in 2019. This was the first comprehensive regional study of microplastics, and we partnered with 5 gyres as well as Chelsea Rockman at the University of Toronto to conduct the study. We had many study objectives. This included improving microplastic monitoring methods, both in the field and in the lab, so it's really exciting to hear all the new development and guidance on analytical methods. Identify baseline conditions of microplastic in the Bay, as well as share our findings and lessons learned widely. So I wanted to thank you for the opportunity to share our lessons learned with you all today. This project was a 3-year study that cost over $1 million. It was largely funded by the Gordon and Betty Moore Foundation, as well as additional supporting funding from these organizations. So I wanted to thank all of them for that. To address our many study objectives, our study design included sampling microplastics throughout the Bay, sampling Bay surface water, prey fish, sediment, bivalves, as well as sampling in the adjacent national marine sanctuaries. We also wanted to understand how microplastics were entering the Bay in order to inform pollution prevention actions. And so we also sampled wastewater effluent and urban stormwater runoff, which in the Bay Area is largely untreated. Our full report, exciting 400 pages of reading, is available on our project website here. And we had many peer-reviewed publications that came out of this project, as well as additional ones that are in progress. This includes a polymer identification method, another one also by Du et al., synthesizing all the different matrices that we sampled in the Bay and did an analysis of that. General lessons learned from this project, which we've adapted that manuscript in this presentation. Some guidance on comparing different sampling methods and QAQC guidance by Hung et al., and a more in-depth analysis of the stormwater results. So I urge you to look at these manuscripts for more details, and my goal in today's presentation is just to give you a high-level overview of our field sampling methods and lessons learned so that you could look for more details in these resources. So I wanted to start with some general considerations for the site selection, and Harry Allen mentioned that it's the importance of site selection in doing an environmental sampling. And particularly for microplastics, this is challenging because collecting and analyzing the samples is very expensive. So our study design was guided by our study objectives. And in order to understand baseline conditions in the Bay, we wanted to make sure we were sampling a good spatial distribution throughout the Bay, including sampling different, say, sub-embayments, sampling closer to microplastics pathways, such as effluent and stormwater discharges, as well as locations further away. We also sampled a reference site far away from the Bay Area so that we could look at compare our concentrations in the Bay with another location that is not so influenced by urban influences. And in terms of sampling different matrices, we wanted to co-locate our sediment sampling and our prey fish sampling to look to see if there were any correlations in the concentrations we observed. We also wanted to consider what were concentrations during dry weather and wet weather. And this was based on our understanding of other contaminants where, particularly in California, where we have a wet season, that can drive stormwater runoff, can take a lot of contaminants into the bay. We got a lot of feedback as we were developing the study design. It was a long process to develop our study design, and even as we were implementing it, we were getting feedback. This included getting feedback from our Bay hydrodynamic modeler as to where we could optimize our sampling sites, talking to Chelsea Brockman, our analytical partner, on how best to collect the sample, what size sample. As Jeff Wagner talked earlier, it's important in terms of the sample size because sampling, analyzing the microplastic samples is really labor intensive, and so you want sufficient particles to get a representative sample, but you also don't want so much that you overwhelm your lab and they can't finish analyzing the samples. We also want to consider how we would analyze the data once we collected it, and we also talked to our QA officer to make sure we would have confidence in the data once we got our results. Of course, we could lay out our best laid plans, but once we're out there, there are logistical concerns. And so our recommendation is to have backup sites because there were times when we went out in the field and we had to adapt on the fly as to where we would collect our samples and what samples to collect. Another important part of the study design is the sampling method. So that's important, and that can change depending on your study objectives. And one important consideration is the sieve size and what size particles you're collecting. This is a graph where we graph the final effluent results reported from various studies. And this graph shows on a log scale the much higher concentrations reported for effluent depending on the sieve size you used. So this makes sense, as Scott talked about earlier with the power law for particle size distribution. As you sample smaller sizes, you'll see more particles. And then I wanted to just reference Hung et al. Who talks, and in this paper we talk further about how the sampling methods matter in the results that you get. QAQC is really important for microplastic sampling. Microplastics are ubiquitous and it is so easy to contaminate your samples. So it's really important to minimize and identify potential contamination sources. And even if you think you've addressed them all, it's still important to take field blanks so that if and when you see, you can try to quantify background contamination levels. We collected both field blanks and lab blanks so that we could differentiate potential contamination from the sample collection versus in the lab processing steps. We also collected field duplicates to see how variable the same sample would be if we collected it at 2 different times at the same location. So next I'm going to just briefly describe our sampling methods for each of these matrices. Just number them so that you could follow as I go through each of the matrices and some brief lessons learned from our sampling. For surface water samples, we use both manta trawls and bulk grab samples. So the manta trawl is like pulling a plankton net across the water surface. So it works kind of like a manta ray skimming the water surface and picking up particles as it's skimming the water surface. This sampled through a large volume of water. The sieve was about 1/3 millimeter. So that's the size limit of the particles that we're capturing. In contrast with the 1 liter grab samples, we can sample a much smaller much smaller particles because we're not sieving them in the field, but it's a much smaller sample. What we learned from the sampling efforts is that the field duplicates can vary widely. In the Mantis 12 samples, the relative percent difference between duplicates ranged from 2% to 100%. And this just emphasized how ephemeral microplastics concentrations can be. For example, there were times where we could see that we were sampling through a foam line or a tidal front that had a bunch of debris and trash in it, and definitely higher microplastic concentrations level. So our recommendation from this experience is to definitely pilot your testing method. It can inform what sample size you should collect. Like I said, you want to optimize you want to capture a representative sample, but at the same time not overwhelm your lab with the number of particles in each sample. It can also help identify contamination sources. As we were sampling, we identified potential contaminations in our field length. In this middle picture, you'll see there was a black mat on the boat, which was a contamination source. And then in our field length, we also saw bright orange distinctive fibers that were probably coming from our bright orange flotation devices. And so from that experience, I recommend collecting blanks from different steps to help identify where contamination might be coming from. And so if you don't get anything else from my presentation, my recommendation is to collect field blanks. I know field blanks are expensive because it's another sample to analyze, but it will give you more confidence in your results. Moving on to sediment. For our sediment sampling, we use a Van Veen brad and use the stainless steel scoop to sample the top 5 centimeters of sediment. This is what we do in the regional monitoring program, and it's more representative of current conditions as well as the layer sediment that is bioavailable. The way we collected our field lengths for sediment samples was to rinse the equipment with reagent water into our sampling equipment. We actually only collected 1 or 2 field lengths, which were actually quite high. So we think that they were a potential overestimate because most of the times when you're collecting your sample isn't touching all of the equipment. And the way to address that is just to collect more field lengths. Another important lesson learned is to record various units of measurement because different studies use different units, and so this is really easy to collect when you're in the field, but if you don't record it, then it's really hard to backtrack that information. So making sure you record wet weight, dry weight, volume, and approximate surface area that you're sampling from in the field. For tissue samples, there aren't any standard methods for collecting field blanks, so we didn't have a good field blank. We didn't have a field blank for tissue samples. For prey fish, we collected top smelt and anchovies, which are abundant in the bay and are important prey species in the bay food web. Our sampling method for the prey fish samples was to collect 10 individual fish at 6 different sites throughout the bay. And our method for the bivalves was slightly different, where at the locations, the tissue was a composite of 3 tissue samples. And so there are pros and cons with these different approaches. If you can get individual species, individual organism data, that's more useful, but then you have more samples to analyze. Some lessons learned is that we saw a large variation in the particle counts for individuals. So for example, for the fish, the distribution was skewed right, where most of the fish had very low particle counts, lower than the average, and there was a much smaller number of fish with much higher particle counts. And so it's really important to collect enough fish, different organisms in order to get a good representative sample. It's also important to be transparent of the fiber counts for comparison with other studies, because as we were trying to compare to other studies, we found that a lot of studies don't report fiber counts because it's so easily contamination source, and therefore need to be able to report microplastic concentrations with and without fiber counts. And we found that lab lengths were extra important for these tissue samples because the lab processing step is very long for digesting the tissue, and that is a potential contamination as your sample is sitting in the lab digesting. For wastewater effluent, we collected final effluent samples, and that was run through a stack of 2 sieves, 125 and 355 sieves. In order to collect a representative weekday sample, we collected a 24-hour composite on a weekday. And our study design was to sample at 4 facilities with secondary treatment and 4 facilities with advanced filtration methods, so in additional to the secondary treatment. We wanted to be able to compare any differences in microplastic concentrations from facilities with these different treatment methods. Some lessons learned from this process is that the conditions at each facility can be very variable. There will be different concentrations in the sample. Your sampling location might be indoors at one site and outdoors at another site, and so the potential for contamination is very different. So it is helpful to have more field lengths again. Even with our 125 sieve size, it was very easy to get clogged over a 24-hour period. And I know this will be an even more challenge if you're sampling much smaller particle sizes. For stormwater runoff, we use a similar sieve size as the wastewater sample so that we could compare these 2 pathways. We used a 125 and 355 micrometer sieve, used an ISCO pump to sample our stormwater location. Our sampling approach is to take sips throughout the storm and then to take a depth integrated sample by tying the end of the ISCO pump to a pole that we could move up and down the water column. So the depth integration is really important so that we could extrapolate our concentrations to potential loads based on flows. This is actually more challenging than we expected. We think we might not have gotten the full depth integration, maybe undersampling the surface water and closer to the sediment bed. There were some sites where there were much higher concentrations, and so it would be helpful to be able to adjust the sample modeling based on what your expected concentration might be at the site. One of our big findings from this study was us identifying abundant black rubbery fragments that we later were able to link to tire wear. And these particles are actually really challenging to identify the polymer with FTIR and Raman, so they may require other analytical methods to sample. And as we did further literature review afterwards, we realized that most tire wear particles are much smaller than the 125 micrometer sieves that we use. And so if you want to get a more representative sample, it would be better to use a smaller sieve size. So I just wanted to summarize my key takeaways from our field sampling effort. Again, do some pilot testing before you collect the samples because you might not know what to expect, and you can adjust your study design accordingly. You can adjust your protocols, refine your methods, refine the sample size that's being collected, and that can help you inform having to adjust your budget needs and time needed for analysis. Sampling may be necessary in terms of particle analysis. We found that when we had some samples that had way too many particles to analyze. And have a backup plan as you're collecting field samples, as you're analyzing samples, and you might meet budget constraints. As I mentioned at the beginning, you can see some of our full report as well as these peer-reviewed publications for additional information. Another recommendation is to have carefully designed field and laboratory templates to help so that to make sure you write down all the important field parameters when you're collecting the sample. We've shared the templates that we use on our project website, so you can go to that project website for all of our microplastic resources. We actually have a webinar that is recorded and posted on our website describing some guidance on study design, analysis, and reporting. So please let me know if you have any questions. This email goes to me as well as my microplastics team at SFEI.
Great, thank you, Diana. It looks like there's a number of questions in the Q&A box. Let's go ahead and have you answer one, and we'll move on and we'll come back to those if we have some time. The first question, were the particle sizes based on actual measurements or just a count of the particle captured in each sieve fraction? It seems like smaller particles could get caught in larger sieve sizes due to accumulated sample debris that are captured along with the microplastic particles.
Yeah, that's a great question. Is this about the graph that I showed?
I believe so.
Yeah, I think the limit is based on the sieve size and not just the particle size. And that's also true for our sampling as well. Like we use the 125 sieve, but yes, smaller particles can get caught in that, and you will get some counts of smaller particles, but your sampling isn't as representative because some of the smaller particles can often escape.
Great. Okay, let's go ahead and move on. And if we have a chance, we'll come back to those other questions. And you can review them as well, Diana, in the Q&A box.